I would like to thank all of you for attending the first BioGeoSCAPE webinar, which focuses on intercalibration efforts. I also would like to thank the many people that have been involved over the years with this BioGeoSCAPE effort, and they're all part of this consortium, and many of them are attending today. So thank you to all. BioGeoSCAPE is the concept for an interdisciplinary global scale microbial biogeochemistry program. The preliminary vision is to understand the function and regulation of ocean metabolism, which we believe is at the heart of Earth's life support system. To do this, we need to elucidate intracellular processes using physiological, biochemical, and omic technologies. And this will allow us to understand community interactions and ecosystem functioning, and then link this to biogeochemical cycles and feedbacks. This schematic illustrates the vision of this program, which requires combining omics and geochemical measurements at a global scale. These data are then processed and integrated, leading to models that could conceptualize and make predictions. This integrated approach will allow us to understand ocean metabolism and its influence on ecosystem health and biogeochemical cycles. This program will involve full-depth basin-scale ocean sections, time series, process studies, as well as laboratory studies, and will result in a wealth of biological and geochemical data sets to improve the integration of observations and models. BioGeoSCAPE is really an international community-driven effort. We started with a meeting in 2018 where we determined international interest. We now have representatives in 23 countries. National planning meetings have occurred in a series of countries are ongoing. We have a good presence in conferences, for example, in the last Ocean Sciences 2022 meeting. And both the US and various countries in the European Union have submitted proposals to organize the launch of the BioGeoSCAPE program at an international level. We are here today because the US National Science Foundation funded an AccelNet grant in collaboration with Ocean Carbon Biochemistry Program to develop at the launching of the BioGeoSCAPE program. AccelNet really aims to accelerate the process of scientific discovery by supporting strategic linkages and coordinating efforts among networks to enhance international multi-team research collaborations. What you see here is a schematic of all the networks that we believe we need to link in order to launch the BioGeoSCAPE program and make it a success. The AccelNet BioGeoSCAPE grant does not support science research activities directly, but instead supports international networking through workshops and educational activities. BioGeoSCAPE will connect with developments in the fields of biology, chemistry, biomedicine, including infrastructure and technology. The study of trace metals, nutrients, and omics in the marine environment is very active, so we will also connect with the oceanographic communities. We view intercalibration and data science initiatives, as well as modeling efforts and rate measurements, as key for the implementation of the program. To bring this program to fruition, and with AccelNet funding, we are planning a series of international activities involving these various domains that I describe here. And these activities are planned for the next 5 years and will involve workshops, meetings, reports, and so on. These international activities are intended to lay the foundation for the BioGeoSCAPE program. This is a proposed timeline for the BioGeoTraces program development. As I mentioned, the first scoping meeting occurred in 2018. Since then, there is various national meetings that have occurred. In the next 3 years or so, we want to focus on intercalibration efforts. We want to talk about data management and infrastructure. And we want to promote all these efforts through workshops, webinars, and so on. At the end of this year, we hope to have the first in-person science plan meeting in the fall of 2023. And during that meeting, we hope to be able to summarize scientific priorities and capabilities of various nations. We also need to forge connections to key supporting administrative structures, and we need to come up with a governance model for the program. We hope by 2025, 2026 to be ready to write some proposals to fund international BioGeoSCAPE expeditions. And hopefully we will be able to launch the program in 2026, as well as the fieldwork. And then in the following 10 years, we hope to release a series of integrated data products. As we move forward, we believe that communication is key, so and networking. So we are working on a new website. We will continue our newsletter, so please sign up in our website. We strive for openness and inclusivity, so we will have rotating leaders and contributors. We want to learn from prior programs, things that work and things that didn't work, and how we are going to do things differently. We believe that this program will be built in a shoestring budget and sort of bootlegging on all other efforts. We want to find ways to create momentum and keep the community going. And we really think that one of the key things for this is to identify heroes that believe in the concept of this program and are willing to put time and effort to help us. We also want to reach out to the younger generation because they are the ones that are to take this program further than we are. So if you want to be involved, please participate in the workshops and contact us. So today we are focusing on the ongoing intercalibration efforts. Producing data that can be effectively compared across space and time is critical for developing a program such as BioGeoTraces. Intercalibration are needed to create interoperational global data sets and will take really multiple iterations. Since we aim to launch BioGeoSCAPE in the mid-century, we really need to make a lot of progress to ensure that when we are ready to sample, we can collect high-quality data. Today, we will hear perspective and ongoing efforts from a variety of communities and encourage brainstorming through small group discussions. We are hoping to inspire further efforts and community building in preparation for the International Intercalibration Review workshop in the coming years, and also the launching of the program in 2026. So with no further ado, I would like to introduce the first talk by Daniel Petras and Jenny Hawkins, who are going to tell us about the efforts with intercalibrating metabolomics. So I will stop sharing.
Okay, hi everyone. I think I'm sharing my screen. Somebody please confirm.
Yes, you are, and we can hear you. Looks great.
Thanks. So I'm Jeff Hawks. I can also be Jenny Hawkins, and I present on behalf of Dan Petras and Carsten Simon, and also the whole group which has been involved in this intercalibration exercise for metabolomics. So it's a large group, so I can't name everyone yet, but I will at the end. The principle was that we wanted to look at how our data sets will be varying if we do LC-MS, MS analysis. So this is a typical metabolomics pipeline. for complex DOM samples. So we chose a fairly simple study design, which was to have a marine extract, which is a typical way of preparing a sample with this PPL sorbent. That was taken from Scripps Pier in La Jolla. And then also an algal lysate, which the intention there was that it would be something complex, which is more of a metabolomics style sample in the sense that it's known primary metabolites. And also ideally something which would be able to be reproduced by others, because it's something we bought from Sigma Aldrich as a powder, which was kind of dissolved and extracted. So we took these two and we made a mixing series, as you can see here. So we labeled them A and M as end members, and then these mixes. With the intention that these 5 samples plus a blank would be analyzed in triplicate by each lab in both positive and negative mode. And that would give us a lot of data which we could analyze in a bunch of different ways in several planned studies. So we're just getting started with the paper preparation. We have a lot of data now, so I'll talk about a bit about that. So it was a 200-liter sample. We wanted to get plenty of stock for the marine, and several internal standards were added to that. So you can see them listed here. Daniel didn't tell me about these when he made them. Several illegal substances in there, so we were happily shipping these around the world. Once I found out I planned to, if I got an email from customs and they said, what's in here, then I would say, well, we don't know, but if you want to analyze it, then you'd be welcome to join our Interlab study. Anyway, we ended up getting 24 data sets. So we sent out, I think, 30 or 32, and 24 labs prepared data sets in time. And so it's a large data set we have. And every lab essentially did the same thing. We had fairly constrained instructions about what you should do. So it was a typical kind of reverse phase separation with liquid chromatography, HPLC or UPLC, and then an analysis with some sort of high resolution mass spectrometer in data dependent analysis mode. So this is where every analyte which is coming through has the potential for triggering a fragmentation event. And then you take this fragmentation pattern and compare it to a library. And these are the settings. I won't go into this, of course, but just to show that we have very well documented settings from every lab. And yeah, the first thing we wanted to do was just to check which labs were finding the internal standards. And so we started analyzing the data with this kind of upset analysis. So we look at the number of labs, 5 found all of them, and so on. So not a great deal of labs, or not a high proportion of the labs found everything. There's good reasons for that. So it could be that it's there in the MS1 scan, but it didn't trigger a data-dependent analysis event, or it did, but the fragmentation pattern didn't match well to the library. So there's good reasons why you might not find internal standards. So we made a cutoff of having to find at least 6 out of 8 to be included in the study. So there's a line here drawn about the ones which didn't find enough. So we started doing some further analysis and looked at how many library IDs they made. So that's matches to known compounds, just clusters. So this is and network nodes. So this is combinations of metabolites found in their data set. So you can see many more peaks were found than those which could be identified to known metabolites. So that's very typical. And we have also feature-based network analysis and extended data analysis technique. So this networking approach allows us to see which peaks are present or absent together in samples. And in total, in the whole data set, we found 27,000 odd nodes, so groups of metabolites, but only, say, 600 library IDs. So only about 2% of the full metabolomics structure in the data in the samples was known or previously measured compounds. So you can see there's a whole world out there which we don't know what it is. When we look at everything which was found by all the labs, we wanted to see how much overlap there was between labs, of course. But we found generally the opposite. So here in this upset analysis, you see that 3,700 peaks were found in one single lab, lab 18. And the next highest node was 1,800 in lab 33. So all of these top sort of 20 intersections were not found in all labs, which was initially a kind of disappointing result. But we realized that the higher peak abundance was, here shown as spectral counts, the more likely it was to be found in more labs. So lab frequency 15 would be found in all labs. And then it's the higher, bigger peaks, which are generally found in all labs. So this kind of makes sense to us. The smaller a peak is, the more likely it is, or the less likely it is to be triggered in every lab as a data-dependent analysis. So we started looking at just the top 100 features. So just taking the top 100 for each lab and seeing how much overlap there was then. And then indeed, 12 peaks, for example, were found in all labs, and that was the highest intersection. And the third subsequent intersections were often found in 13 or 14 labs. So the data sets start to align as you look at only the highest features. And again, if you look at the top 20 features, then yeah, mostly they're found in every lab. So there's this high amount of overlap when we look at the most abundant features, but as we go into the thousands of features that we find in each data set, we start to get more and more unique features.
Jeffrey, you're a little over time. Can you finish up?
Sure. Yeah.
Thank you.
So very simply, then we did a principal component analysis. We find good clustering between the A and M samples, although we find that the M sample is more different between sites, which is what you would expect because it's a more diverse sample. And so the take-home messages are here. I can share this later so you can see it. But basically, we're just scratching the surface, and it's really important. We found that the partners actually followed the instructions. That was the main reason why labs didn't find the internal standards. Very quickly, I just want to mention that we have a reference material which we can share. So I can share information about that later. Thank you for your attention.
Thank you so much, Jeffrey. All right, so I would like to introduce the next speaker. It's Adrian Marchetti, who is going to tell us about the US nucleic acid intercalibration efforts.
Hey, thank you, Maite. Can everybody hear me okay? Okay, I'm assuming that's a yes. So I'm just on behalf of the organizing committee of this workshop that took place in January of 2020. Essentially, what we were trying to do is build a roadmap towards community-wide intercalibration and standardization of ocean nucleic acid omic measurements. So in terms of what we're referring to when we talk about nucleic acid omics, the focus was on sort of these three approaches. So looking at amplicon sequencing, mainly with a focus on 18S and 16S rDNA, metagenomics, as well as metatranscriptomics. And the primary focus organisms were bacteria and archaea, so heterotrophic and autotrophic, as well as protists, again, heterotrophic and autotrophic. And so essentially what our research objectives are to find key sources of variability in the NA-omics approaches, essentially to enable measuring and monitoring of the ocean through NA-omics in a standardized way, to provide data sets conducive to scalable computing and modeling, and to train the next generation of scientists in the implementation and standardization of NA-omic approaches. So with that, obviously there's a question in terms of why now? And essentially, we know that biological oceanography has now entered the molecular era. So there's a lot of labs out there doing various approaches, and quite often they're left to sort of come up with their own protocols and own techniques in how to do these. And so then sequencing itself is a very common way in which now we are interrogating the ocean. And even though we know it's a moving target, the sequencing landscape, at least over the last decade or so, has become fairly stable. So that is providing for a good opportunity in relation to doing these sort of efforts. And there is yet to be a coordinated standardization and intercalibration effort towards community-wide best practices and intercomparison. And, you know, given we have programs like BioGeoSCAPES on the horizon and ones that have already been implemented, we feel the timing is right to actually come to do these sort of standardization intercalibration efforts. And then lastly, this would enable scientists to lead the charge in a community-based omics program. So a lot of these techniques are basically used in other disciplines, and we can lead the charge in this regard. So with that, we had a workshop, as I said here, at UNC in January of 2020, which was funded by OCB and partially supported. Sorry, this is going on its own. Partially supported by the Simons Foundation and NASA. And the organizing committee included myself, Bethany Jenkins, Andrew Allen, Paul Berube, Scott Gifford, and Allison Santoro. And we had 32 participants, all of different ranks and specializations with various levels of experience using the NAomics techniques. So the workshop was over 3 days. And essentially, if you're interested in the workshop report, it's available at this website. But what did we do at this workshop? Essentially, the overall goal was to enable the analysis, reuse, and long-term impact of harmonized and quantitative NAomics data. So essentially, the goal is here, if we have different transects in the ocean and different labs analyzing or extracting sequences, we can do so in a way that they're all intercomparable. And so during the workshop, we identified potential sources of variability and bias for NAomics data production pipelines. We prioritized potential sources for assessment. We created a plan, essentially, for the research activities to provide empirical data on variability and bias during NAomic approaches. And then we developed a framework for community-wide intercomparison. And so what I'm just showing here is just a table that was provided in the workshop. So you can see here some of the major categories that we're looking at include sample collection, sample filtration and storage, internal, external standards, DNA and RNA extraction, sequencing, and sequencing analysis. And each of these approaches have various levels of variability in terms of the different sort of factors that may be introducing error that we would like to address during this project. And so just lastly, since this workshop, we've been collaborating extensively with other programs, including BioGeoSCAPES, providing workshops or providing presentations at workshops, both for the US BioGeoSCAPES and as well as international efforts. We've given a presentation and been in communication with folks from Atlantico who are also very interested in these sort of approaches for NAomics. And many of us have been working with exports as a nice way because of essentially having actual data sets that we can intercompare to start these efforts. So again, this is the website here where the workshop report is available. I encourage you to go take a look at it and would be happy to answer any questions.
Thank you, Adrian. So for the sake of time, we're going to go to the next talk. It's by Mark Saito on the efforts associated with intercalibrating metaproteomics. So go ahead, Mark.
Great. Thanks, Maite. It's great to see everyone here. This is really exciting to get together like this. There's been increasing interest in proteins in the environment and measuring them directly because they can provide a functional perspective on microbial biogeochemistry, being structural components or transport components or catalytic components of cellular machinery. Let's see if my slide will advance. There it goes. So a few years ago, the ocean metaproteomes community self-organized to launch an intercomparison effort, and I'm representing the participants of the Ocean Metaproteome Intercomparison Consortium. Here's a picture of one of our workshops and a list of authors. We have 15 institutions involved. This is an OCB activity, and OCB provided funds for the workshop and sample shipping. The sampling was bootlegged on a previously funded cruise, and metagenomic analyses that we needed to do our protein analyses was donated by the Naval Research Laboratory. And labs basically volunteered their sample analysis of 1 to 2 samples. And so this study design basically involved collection of samples from the Bermuda Atlantic Time Series study, where we used McLean pumps and the AUV Clio. In this case, we had 2 pumps clamped together with 4 heads, resulting in these 4 142 millimeter filters, and they were sliced into eighths and distributed. And then we had 2 arms of the study. This was a blinded study, so it was anonymous submissions. And we had a laboratory component where we stipulated one-dimensional chromatography. So not necessarily our best methods, but a method that everybody could do that would be reproducible across all laboratories. And users submitted both raw data and process data. And then we had an informatic arm as well, where we, after the laboratory component was done, we then shared files from Bermuda again, and then let labs compare their informatic pipelines. And the results were pretty promising in terms of consistency. We saw consistency in terms of our taxonomic groupings, our functional groupings, and our quantitation. Here's the taxonomic representation where these are major taxa shown here across triplicate analyses from 7 laboratories. And you can see the patterns are pretty reproducible across laboratories. We saw similar things on the functional level that I don't have time to talk about today. But then also on the quantitative level, when we did pairwise analysis between these laboratories, this line here, the diagonal line is unison between the single laboratory, but then you can see pairwise analyses between the other 6 laboratories. And in general, you see a positive correlation, and indeed the linear correlations across these laboratories all had R-squareds of higher than 0.5, which I think is pretty good for these complex omic data sets. There was good coherence of peptides between laboratories. This is a Venn diagram between 3 laboratories showing about 3,000 of the peptide constituents of proteins shared between these 3 groups. But there was a lot of variability in depth of analysis and stochastic detection of rare peptides. Like Jeff was talking about from the metabolites, there's a lot of features, a lot of proteins and peptides in these, and we're not necessarily detecting all of them, but what we are detecting seems to be quite reproducible. And so our status and lessons learned include consistent results across all laboratories. Yet the depth varied, as I mentioned. Here's a similar intersection plot showing proteins that were consistent across all the laboratories, but sort of a long tail of rare proteins that vary between laboratories based on just the complex spectra and how the instrumentation detected them. We have a manuscript drafted. We just sent around the second draft of it yesterday, so hopefully we'll submit it in the next month. Some lessons learned were trying to stay focused on short-term goals and trying to keep the scale nimble and manageable so we could keep our momentum up. We piggybacked our sample collection, so it's largely a volunteer effort with no large proposals so far, except we had OCB support for our workshop and our shipping, but not for analyses or salaries. We actually de-scoped our study from 3 parameters to 1 parameter, leaving the 2 other future studies once we realized we had our hands full. In general, though, this was a really positive community building experience, I think. I met a lot of people, and I think a lot of us met people in the ocean metaproteomics community, as well as the international metaproteomics and microbiome community from soil and health fields as well. So that was really exciting to connect and get to sort of brainstorm with that community. And I think the community is interested in follow-up efforts. We're going to hopefully talk about that in the future, potentially DIA or targeted proteomics next, including absolute quantitation, which would be really exciting to do. And I just want to spend a minute now also offering future intercomparison particulate biomass sample sets for this BioGeoSCAPES community. We have a cruise coming up. Alison Santoro and I are co-chief scientists of a cruise to the Eastern Tropical North Pacific, and the Clio autonomous underwater vehicle is coming out that is capable of these large volume samples. So we'd like to offer up batches of samples, say 16 to 32 samples per batch, depending on how it's sliced, to 2 to 4 groups or something like that that's self-organized. So we'll try and get that announced, but feel free to contact us if you want more information about this. Thank you very much. Great to see everyone.
Thank you so much, Mark. Our next talk is a joint talk by Luke Thompson, Alice Larkin, and Harriet Alexander, and it's about BioGeoSCAPES nucleic acid intercomparison.
Great. When I try to share my screen, it says disabled share. Okay. Is there a way I can be a presenter?
Yes.
You should be okay now.
I got it. Can you guys see that?
Yes.
Great. So yeah, my name is Luke Thompson, and I'm representing the BioGeoSCAPES team, in particular on this exercise, Alice Larkin and Harriet Alexander. And so here's a picture of all the BioGeoSCAPE team members. And so just a brief background on BioGeoSCAPE. So the GeoSCAPE set of cruises is a decadal scale oceanographic survey of primarily physical and chemical oceanography of the global ocean. And it's a wonderful platform for exploring the ocean, but at the moment there isn't a sustained biological observation system as part of GeoSCAPE. And so that's what we want to do. So to develop a deeper understanding of the link between those physical and chemical environmental parameters and the diversity of global ocean plankton and their biogeochemical roles. So there's a number of different observations that we're doing with BioGeoSCAPE, including inline as well as CTD, and then post-cruise and analyses. And today I'm just going to focus on the nucleic acid part, the eDNA and metagenomics. So the basic workflow of any kind of environmental survey, and shown here on the bottom is my former postdoc Sean, who's on the Ron Brown, one of the GeoSCAPE ships, taking samples from the CTD. So we use SteriVex filters. That's kind of been decided, but the question is, how do we preserve and extract the DNA from those SteriVex to get well, there's a number of things we're trying to optimize. We're trying to optimize the quality of the preservation, also, and that is shown in the quality of the DNA that we get out the other end, as well as efficiency of extraction, because we're dealing with hundreds of samples, potentially several hundred samples a year, and we want to be able to extract those in an efficient way, and also have a sample preserved such that if something goes wrong, you lose a freezer or something, that it's still available to be used. So what we're testing at the moment is some different ways to extract from the SteriVex, shown in the middle bottom here. One of the ideas that we're pursuing, or methods that we're pursuing, is using beads inside the SteriVex to do the extraction, and in some cases adding the beads prior to filtration to really speed up the extraction process. So there's all kinds of material in the seawater, of course, and we want to extract that. So that's just what this is showing. So there's been 2 intercalibration exercises that we've undertaken. We're still waiting to sequence all these together, so we don't have a lot of data yet, but I'll describe the plan or the experiment that we've done. So one is using samples that were preserved on the Pacific cruise PO2. They were all preserved in the same way, using a lysis buffer that's been used by Adam Martini's lab at UC Irvine. And so we're extracting that in 3 different ways. In cartridge with heating and lysozyme is the first method. At the Martini lab, second method by Harriet Alexander's lab is excising the filter and then bead beating. And the third method is in my lab at AOML in Miami, and we're doing in cartridge bead beating and then using this Zymo kit with the Kingfisher robot. And so, however, there's a couple other methods. I mentioned that bead beating, where the beads are added prior to filtering, and this really would save us time because it's quite a bit of work. It works really well to use bead beating inside the SteriVex, but it's quite a bit of work to get those in once there's liquid of some kind in there. And so if we can add the beads prior to filtration, that could really save us a lot of time, and also allow us to use a preservative that's left in there. So we've got 5 methods we're testing in this second set of tests, and I just got the filters today from Elise. And so we've got their standard method that's been used in the past, and then a method that we're planning to use for samples that have already been collected. Third would be the method that we're use at AOML, where we don't have any preservative in the SteriVex. And then 4 and 5 are either a 1x or a 0.5x of Zymo's DNA RNA shield. And so this will really cover all the bases and allow us to know whether if we do change methods that we're backward compatible with different methods, including ones that we've used. So that's all I have, and I'll pass it on to the next speaker.
Thank you so much, Luke. Our next speaker is Daniel Giudicono. He's going to tell us about intercalibration perspective from Atlantico and Tara programs.
So hello everyone, yes, Daniele Giudicono from Italy, Naples. I'm the coordinator of a large-scale project on the microbiome of the Atlantic Ocean. And I will quickly remind, first of all, the project is a large-scale project, so 36 partners, it's EU funded. And the aim is also to establish a strong cooperation with Brazil and South Africa, which is a bit specific of this project. Here, I just want to remind that we are having a series of cruises using protocols, including omics, including also proteomics and metabolomics. So basically, we are implementing already BioGeoSCAPES in a way. And a part of the flagship cruises, we are spreading the protocols all over the Atlantic with many other partners. We are collecting a lot of data on this kind. But most importantly, our aim is also to develop cooperation, deep cooperation. So we are established a relationship with a series of sequencing centers and the research centers in Brazil and South Africa to organize a biobanking locally, local analysis, but also to develop local empowerment. So basically, one of the exercises we are doing is an intercalibration of the sequencing centers for the analysis of data in the future. The final aim is to support Brazil and South Africa also to develop in the future regional, let's say, organization of groups working together on these fields. Very quickly, what we are organizing is an intercomparison specifically for the omics, so metabarcoding, metagenomics, metatranscriptomics is basically doing a size fractionation over 3 sizes, so going from CO2 for viruses up to 20, 30 micron. And once we have this size fractioning and preservation into liquid nitrogen, we're actually, we are applying this approach to a pond, so it's fresh water that this exists close to the French sequencing center, the GeoSCAPES. So we organized the filtration there, and we split the filters into 4 parts, which should be stored separately. And what we are doing now is to actually spread the filters over 3 sequencing centers. So we have 3 replicates for meta B for each size fraction. We have 3 replicates for meta B, for meta G, meta T. We have a control with millicool water, so basically distilled water. And at the moment, we are sharing this between GeoSCAPES in Paris, Cape Town, University of Cape Town, and Sao Paulo in Brazil. And we are 1/4 quarter, which is actually available if anyone wants to contribute to this exercise within BioGeoSCAPES. The exercise consists in the full RNA DNA extraction, the quantification and quality assessment, and the full sequencing. Overall, I want to remind that all our protocols and the handbook on standards and best practices on omics and imaging are available and are on Zenodo. And if anyone wants to know more, there will be in the breakout session Stéphane Pezon and other experts, Emma Rocker from South Africa, Pedro Oliveira from Brazil, and others that will be available to further discuss this collaboration. Thank you very much.
Thank you so much, Daniele. All right, we're up to our last speaker, but not least. This is Julie Laroche, and she's going to tell us about rate measurements intercalibration efforts. So, Julie, are you there?
Julie, you are muted if you are speaking. Can you start over for us? Thank you.
Can you also go back to your first slide, Julie?
Sure.
Please.
Okay, am I okay now? Yes.
Yep, you sound great now. Thank you.
Okay. Yeah, so the first slide, there's nothing on the first slide. It was just this here. Okay, so I was asked by the organizers to talk about our experience with the natural fixation measurements more than a decade ago. And for full disclosure, Vipka Moore takes all the credit for this work. So Vipka, who was then a PhD student with me and in Kiel, was doing some lab experiments with Procosphaera and measuring nitrogen fixation rates. She identified that the method as used underestimated quite significantly the true nitrogen fixation rates in her cultures. And so building on these lab experiments that she did, and then the problem that she had identified, we carried out field studies and compared some methods in the context of other oceanographic data. So you can see, if you want to have a view of what these papers were about, they are posted here. So it's essentially, more than also a decade later, there's been a more recent paper by a large group of people, of researchers, that looked deeply into many aspects of the method and that reviewed all the potential errors that could originate from different aspects of how you carry it out. The reason I'm showing this then is to go to some lessons learned, and I have two. And so the reason that Vipka was able to identify the problem was because she was doing a mass balance in her culture. So trying to account for the growth, the biomass increase based on the nitrogen fixation rates, and then they didn't add up. So that's so the ability to carry out the mass balance was the key to identifying the problem in this case. Of course, it's not always possible to have pure cultures depending on the rates that you are measuring. And so the second lessons learned here is that when you assess a new method, it's important to like if you do correlation between two methods, it's important to be aware of that this can be tricky and that can be misleading. So what happened when this, years ago, when this 15N2 method was designed, there was no marine cyanobacteria culture, and the 15N2 method was calibrated with the acetylene reduction assay, which is another way of measuring nitrogen fixation. And the ARA requires a conversion factor to relate back to nitrogen fixation, and then that's usually measured by the stable isotope method, the 15N. And so it's important to, if you need a conversion factor to relate back to what you're trying to measure, then it's important to be aware that that could lead to some problems. And also with the isolation of new cyanobacteria to carry out these measurements in the lab, it's also important to remain critical about updating and testing your old method and then new methods. So the final thing that I want to discuss here is some thoughts on how we could go forward for doing this rate measurement intercalibration. And this is, I guess, not as advanced as the other aspects of BioGeoSCAPES. But I think if you have a new method, try to first test it on relevant culture organism and against an old method. And to do that, you would maybe design a way to assign appropriate specific organisms for the calibration of different processes. And also that might require the isolation of more type strains as reference material for rate measurement. So the complexity in measuring your rate is like orders of magnitude greater than measuring a concentration of a compound or an element in the ocean, clearly, because you need the live material. You can't easily preserve it while you're doing the measurements. So understanding that it's difficult to obtain these strains, so it might not be always possible. So maybe the next best thing would be to establish time series sites as designated platforms for intercalibration of rate measurements. And this would be because it's easy to access these time series, and then there's also a long history of databases for intercomparison. So like to maintain the internal consistency of the results and the rates that are supposed to be roughly order of magnitude. Yeah. Okay, and then so so maintain a suite of core rates measurements, and then at these selected sites, and also link the new methods to old methods. And then there are a lot of new methods coming and now with single cell measurements and so on. And then this would be a way to also for the community to have access to these various platforms continuously throughout the project. So that's all I have.
Thank you so much, Julie. So I would like to thank all the speakers for very engaging and super interesting talks. And now what we're going to do is move to the discussion part of the webinar. And I would like to pass the baton to Harriet Alexander.
Thank you so much, Maite. All right. Can you see my screen? Yep. Okay.
Yes,
so we're going to enter breakout rooms to carry on the discussion. Thank you so much to all of our speakers for a really wonderful overview of what sort of efforts are going on in the different communities. We have set up jam boards, which I think by now many, if not most of you, have experience with in these types of meetings. It's an interactive kind of web board where you can post different little post-it notes with thoughts. So we've created a jam board for each of the groups that we're about to break up into. We're going to have links in the chat for each of the jam boards. So once you're assigned a group, go and find which jam board you should join. Ideally join before you enter your breakout room, because I don't think you have access to the chat once you're in your sub breakout room. To wrap up by 12:30, and when we come back to the main group, we'd like one reporter from each group to type the 3 main takeaways from your group's discussion for your group. We'll definitely make it so that we can distribute the Jamboards themselves. Also, one other note is we've kind of preloaded the Jamboard with the questions as well as the main little subpoints. But if you need to overflow, there should be another slide afterwards that you can kind of move on to if need be. So feel free to add as many thoughts as you have. And just to quickly go over the questions, the first question that we want people to kind of target is, what are the greatest challenges limiting intercalibration now? Here are a couple different points, but do we have sufficient heroes to organize the efforts? How can we really democratize or engage the full community in these efforts? What about standard sample availability, resources, financial and otherwise, or any other challenges that you can think of? And then we want to think a little bit about moving towards this international intercalibration workshop that we're planning for in the next coming years. So what do we need to do to really prepare for this intercalibration workshop? What do we want to accomplish at the meeting? When should we try to schedule this meeting? How far out in our timeline? And then a couple other questions more broadly. How can we create and distribute standards? Of course, this is going to be kind of dependent on different types of measurements that we're taking, but a consideration to keep in mind. And what can we do to help you facilitate intercalibration activities in your community? Again, the ExcelNet is this idea of bringing networks together. And so there are a lot of smaller networks. The BioGeoSCAPES example is one of them. These smaller scale intercalibrations are just as important as our kind of larger community ones. So how can we help you do that in your community? And also, what can we learn from other communities? We know that the chemists amongst us have done a really nice job of running these kinds of intercalibration exercises. So with that, we're going to go into breakout rooms. And I'm going to turn it over to Heather. And oh, there we go. You should now see the room that you're joining. And if you look in the chat, you should see that there are Jamboards for the different group members. So ideally, go to the Jamboard that the group you are supposed to be in and enjoy.
Okay, so I'm going to share a poll I'm going to share two polls, but I'm going to start with the first one here, and I will share my screen so you can watch the results come in real time, but these kind of springboard off of the discussions that you just had in the breakout rooms. So stand by. So you're going to go to menti.com and enter this code, and then you should be able to enter responses. This is the first question. Our responses are slowing down, so please take the last couple seconds here to get your response in before I move to the next question. How ready are we for this International Intercalibration Workshop? Okay, I'm moving on to the next question. It looks like we've got 1 to 2 years seems to be the leading answer here. And our second question, and this is a word cloud, so just enter short phrases or individual words, please. You should be able to submit up to 3 responses, so you can put in more than one. Another couple seconds. All right, well, there you have it. It looks like standards are definitely are jumping right out at me. Standards, reference materials, parameters, data, community building. And we'll plan to share the results of these polls in addition to sharing the recordings from today. So now I have a second poll for you all. Let me just get it open up on my screen here. Okay. So this next one is a ranking question. Getting a little more sophisticated with our mentees here. So there's a new code. This is a separate Menti poll. So please use this code from the Menti site. What do you perceive to be the greatest barriers to intercalibration at present? And this might vary by country. This could vary by community, by omics groups. If you have other, please do specify in the chat. Just say other equals so that we can get some of that conversation going too. Funding in the lead. Oh, we need champions and we need funding. And I can't see the chat right now, but I hope people are elaborating a little bit in the chat. Couple more seconds on this question, and then I'm going to move to the next one. Hopefully Mac and Allison's upcoming cruise can help alleviate sample collection and distribution a little bit for a few of you folks. All right, moving to the next question. So this is just short answer. These will come up as speech bubbles. So how can we sustain international community engagement and forward progress on intercalibration throughout the duration of this CellNet? We want your ideas. We think this is one of the foundational elements of BioGeoSCAPES, and so we anticipate that intercalibration will have, webinars and perhaps a small group of champions to help build international community around intercalibration activities. And we do have this international workshop that will happen at some point during the CellNet. So this is important to us, getting your feedback on how we can make sure to sustain the momentum and keep community building going throughout this entire CellNet. This is an important part of BioGeoSCAPES. I'm going to try to scroll through these slowly so you have a chance to look at them. Keep them coming. And as I said, we are going to make a recording of today's webinar, all of the results from the polls, all of the Jamboards. We will make slides available from Maite's introductory slides. and our closing slides available. If you would like access to our speaker slides from today, please just reach out to the speakers. Just another couple seconds to wrap up our poll. Great. Thank you everyone so much for your input on this today. This is exactly what we need, and we need to keep doing it throughout the duration of this ExcelNet activity. So I will stop sharing and open the floor for some more open discussion.
So I can jump in. We've basically had, some short talks and some nice focused small group discussion, but we thought we'd spend a final, few minutes here allowing people to speak across the whole group if they have any thoughts they want to share. And looks like Daniele already has his hand up. Would you like to say something, Daniele?
Very quick comment. During our experience with the quiz we organized with Tara in the South Atlantic, it was really useful to be confronted with very different realities and situations depending on the countries. So there is a huge gradient between US, EU, and other countries, and I think we should really keep this in mind in terms of designing the protocols, but also any kind of exercise like the intercomparisons like this should be designed also keeping in mind the challenges that sometimes there is not people trained in some countries. So training could be a capacity building should go together with these kind of exercises, for example. Or there are no sequencing centers which are, or other things which are ready to treat environmental samples, et cetera, et cetera. Just to say that it was a very good lesson for us to confront, and I think we should now on keep in mind. I have other colleagues here from the project, so Paula from Brazil and Emma from South Africa. I don't know if you want to comment on that, but please let's keep in mind if you want to have a really global participation. There are challenges we do not normally consider.
Thank you. That's a great point. I think that the hope is that BioGeoSCAPES could be very international, very much like you've modeled in Atlantico, and we could collaborate with countries all over the world and involve them in their and help them study their coastal environments as well. So yeah, I'd love to hear more perspectives on that or any other comments.
Yeah, I can probably follow up on that a little bit of what Daniel said, which is absolutely true. Here in South Africa, we discussed this in our breakout group as well. There's been extra challenges. So those of you most people work with Illumina. In this country, the consumables, we get dinged with a huge fee every time consumables get imported into the country. And we found the more sort of noise that we make, we've got this big consortium that's attempting to do this big data set. And companies such as Oxford Nanopore and BGI have actually been quite cooperative in this respect. And it's opened the eyes of Illumina, and they're starting to pay attention a little bit. So it's been an educational exercise in that respect as well. And as Daniel said, there are challenges this side that just don't exist in the north. So the more people we can get from the south, even better.
Thanks for that comment. Any more thoughts along those lines or others?
I think maybe there's models out there to help, and it's kind of slowly building up, but I think there's a strong possibility to get there. For instance, I know there's been development of a global ocean acidification in a box program, which provides for about 24,000 a whole carbon chemistry kit with pH, alkalinity, a CTD combined with data analysis and reporting and best practices videos with subtitles in Spanish and Portuguese and French. And so there's these models out there of how it's been done, and how we can think about doing it and applying those lessons learned from other, parts of oceanography, chemical oceanography, for instance, and think about applying those models here. I think it's the same thing with, there's a lot of interest with standards, for instance, on the biological side of things. And, for a long time, right, there's been the carbon certified reference materials that were produced by just the Dixon lab at Scripps, but we're now working to transfer that to NCEI, which is the Standards Institute here in the US, and provide those for the world. And so I think we can think about applying those models to the biological component, and there's strong history there that we can really take advantage of in accomplishing those goals.
Can I say something? I think one thing which is a little bit unclear for me is how big the community is for metabolomics, for example. Like, obviously, metabolomics itself is enormous, but for specifically, aquatic or marine metabolomics, we kind of have an impression. The people here, we know roughly who's doing what, who's publishing papers, but there may be way more people who would be interested in doing this kind of analysis. And so when it comes to things like preparing reference materials, you don't really know how much you need to make, right? Until people start ordering it, using it, and then you find out you've made, 10 times too little or 10 times too much, put too much effort into it. So I don't know what the solution to that is, but we need some sort of communication via conferences or something to find out, to allow people to sign up, right? I want some of that if you make it. And that obviously includes the Global South as well. And it's totally different if, North American, European labs can pay for something where perhaps in Latin America or somewhere they can't pay as much. So we need to know where people are, how many they are, and how much they want. And currently it's really difficult to access that information.
Go ahead, Tim.
Can I react quickly? Yeah, I completely agree with you, and just want to underline that there is now the UN program. It's called OBON, on biomolecular observations. It's trying to serve as a way to coordinate globally this kind of efforts in terms of sharing practices and etc., but also to collect, who is working on the field. Let's say, I think what is really nice with BioGeoSCAPES is that this is a scientific program, and this is the only one that I know at a global scale. So there are a lot of efforts to coordinate, but there is, I don't see any other alternative, any place where there is a discussion on science. So I think maybe BioGeoSCAPES should also serve as a way to collect, to make an inventory of who is doing what on these fields globally. I think that would be useful. I agree with that, or would be interested.
Thanks. Those are really great points. Reaching out to more people and new communities who may be interested in making these measurements is really key, I think. And one thing we saw something similar in geotraces where people were new to measurements and actually used the intercalibration as a way to sort of better their laboratory capabilities and learn how to do methods through the intercalibration process. So that was actually, I think a lot of people don't realize that, but it was actually a really great way to entrain new countries actually through the intercalibration, because we could share these samples and say, hey, look, and then once laboratories got numbers that were similar to other numbers, then they had a lot of confidence that they were ready to participate. maybe and reaching people is a good point. I mean, we have this email list, but which is quite international now. So as people are embarking on new studies, they could, advertise them for participants as samples allow. I think that may be the challenge, right? Any more comments and thoughts? I know it's a little bit more. Oh, Kay's got his hand up. Go ahead, Kay.
Thank you. I just thought the common database could be useful, especially if it's in an easy way to use. And personally, as a modeler, I'm hoping to compare the modeling result with the dataset, great dataset observationalist experimenters provide. And the challenge I'm facing may be where to find these and how to download it and so forth. And easy interface would be really useful, and excited to contribute to the overall project as a modeler.
Thanks, Kay. Go ahead, Maite.
Thank you.
Yeah, in our group, one idea that came up is how important it is to have the different communities talking to each other, like we did today, because today, for example, we discovered some common findings in different intercalibration efforts, which I really think could help us focus intercalibration efforts. So I think having also these communities talking to each other is important, not just within your community.
Thanks, Maite. Just got a couple minutes left. Jackie, would you go ahead?
Yeah, so our group also discussed a little bit kind of tangentially about how it could be needed to, building off what Kay said, follow up on metadata standards across the different groups. So even between the individual disciplines, so metabolomics, proteomics, genomics, but also internationally. So that way the metadata is curated and mappable, and maybe advocating for that as an important initiative that needs to be done within this broader scope. So that way all of our data will work together instead of being siloed by various nationalities or subdisciplines.
Thanks, Jackie. I see we have a comment from Ben as well. Ben, do you want to say something about it, or want me to read it? Oh, can't hear you. No luck. I can read it then. Let's see. Ben says, having standard benchmark data sets is huge to bring together non-domain experts, not oceans, but some are thrusts related to that comment. And there's a link to proteomicsml.org. Any last comment before we wind up? I've got a closing slide I can present when we're all set. Anyone want the last word?
I'll have the last word. I just wanted to say that every task seems huge until you kind of start biting at it. How do you eat an elephant one bite at a time? That's always something to remember.
Thanks, Martha. That's great. I don't want to eat an elephant though. All right, let me share the closing slide. There's nothing else? Okay. So on behalf of the team organizing this webinar, Harriet Alexander, Maite Maldonado, Heather Benway, and May McKeegan, and myself, as well as the BioGeoSCAPES consortium, which is all of you and the organizing group that we have with the AccelNet and the effort so far. We want to thank you for engaging on this important topic. If we're really going to try to understand the changing metabolism and nutrient cycles of the ocean, we're going to really require a global intercalibrated effort. To understand differences across space and time is going to require measurements to be intercomparable, and that's going to require intercalibration. As we've heard today, that's going to necessitate organizing and laboratory work to be accomplished prior to launching a BioGeoSCAPES program. But as we also seen today, there's a lot of positive community building and interesting science that can be accomplished simultaneously as we deepen our understanding about the reproducibility and accuracy of our various exciting measurement capabilities. So we'd like to stress that BioGeoSCAPES community aims to be inclusive and open. And if you want to be part of these efforts, we're always looking for heroes to engage with communities. And thank you for participating in this effort. There's a lot of great information captured here. Feel free to reach out to one of the organizers here. And we want to have people be aware of future activities. So we're hoping to launch another webinar later this spring on data management and informatics. Again, sort of has efforts of organizing and pre-work and networking in anticipation of a real in-person meeting that we'll fund and have occur again in the future, like the intercalibration meeting. We're also hoping to have an international science planning meeting in the fall. We're trying to start getting that organized. We're going to start rolling out educational and early career opportunities such as inter-lab exchanges and things like that we have financial support for through this NSF SLNet grant. And if you haven't already, please sign up for the biogeoscapes.org. And with that, thank you, everybody.
Thank you all.
We'll see you again soon.
Thank you. Bye. Thank you for organizing.
Thank you.
Can you pause the recording, Heather?